The 41st Annual National Water Well Association Convention and Exposition was held in Houston, Texas, October 29th through November 1st, 1989. A highlight of the annual convention is the keynote presentation given by the Darcy Lecturer. The Henry Darcy Distinguished Lecturer Program is sponsored by the Association of Groundwater Scientists and Engineers. AGWISI annually chooses an individual to be offered as a visiting lecturer to a number of universities in North America. This program honors the historic discovery by Henry Darcy in 1856, which established the physical basis on which groundwater hydrology has been studied for more than a century. In 1977, Randy Bassett received his Ph.D. in environmental geochemistry from Stanford University. He then joined the USGS Water Resources Division as a research hydrologist, while also being a research scientist at the University of Texas Bureau of Economic Geology. In 1982, he entered the private sector as an exploration and research scientist for Stauffer Chemical Company. After a corporate merger, he began his own research consulting firm, Geosystems Research. Dr. Bassett joined the faculty of the University of Arizona, Department of Hydrology, as an associate professor. Dr. Bassett's focus will be the utility of chemical modeling with and without advection in natural and environmentally stressed hydrologic systems.
I thought I'd begin with a slide of the University of Arizona campus. This was a very popular slide when I was up in the Northeast when it was cold and snowing outside. So I would like to invite you to come to our campus. Many of you are familiar with the Department of Hydrology there, and we are very proud of our campus and very much interested in exchange of information as people come through and want to discuss this whole concept of hydrology with us. I also want to recognize the fact that NWWA does sponsor this, and I appreciate very much their allowing me to travel from place to place. There's the title, goes on for about 4 lines. So I won't give you time to read the entire title. What I'd like to do is begin with a diagram. This is a facsimile of Darcy's original experimental apparatus. Now, you may be familiar with Henry Darcy. Henry Darcy lived in the mid part of the 1800s. And we're all familiar, of course, with the results of his experimental work. He was a civil engineer who lived in Dijon, France. And one of the things that I've discovered in traveling around as they pass out the condiments for the sandwiches that you get on airplanes is that mustard package also comes from Dijon. I suspect Darcy dealt with that very same burning issue that we deal with, is whether or not we have any poupon mustard. But one of the things I would like to mention about Darcy, and of course that is the title of the series, is that he was a very precise and careful engineer. And as a result of his work, the documentation that he did, the very precise experimental work that he did, which resulted in the presentation of his law, it serves as a springboard for some comments that I would like to make today about this whole concept of error and uncertainty. Because he was very careful in his experimental work, we were able to derive this mathematical formulation, which is so important to us in our business. And I think this is a concern that we have in the university environment and where we find ourselves, as well in the profession where we do a lot of our work. And this whole concept of the chemical data being the central part of much of what we do. Now, I want to talk about the chemical data briefly. There are 3 aspects related to that chemical data. There's this collection procedure that we have to deal with, the analytical procedures, and of course the interpretation. Now, I want to spend a very little portion of my time talking about the first 2 issues, and the last part of it dealing with the interpretation. But I do want to make some comments about the error aspects of the chemical data. You see, we are spending an awful lot of money, as you know, characterizing our sites, doing the preliminary work. On my final results on the back of a paper towel and putting that in my lab book later in the day, and I wasn't always so concerned about the proper documentation of all the experimental work that I was doing. And I think more than any other time, hydrologists and geologists are having to deal with this issue of their data being scrutinized. You know, we're in a business now where the data that we collect are not going to just sit on the shelf. Very often they're going to result in some kind of litigation activity, and someone's going to review your procedures. And one of the things that seems to be happening all across our nation is that there are many consulting firms that have emerged whose sole responsibility is to review what you've done. And so you're sitting there on the witness stand, or you're giving your deposition, and one of the things that they're going to be asking you is, did you follow that specific EPA protocol? Are you sure that you've documented every aspect of what you've done? And if you have not done that, then there is a strong possibility that your data will become inadmissible. So this aspect of review and scrutiny is a part of our entire business that we've not really dealt with in a long time. I kid with some of the other fellows here about our publications list and how many, my reader-author ratio I think is 2 on most of the publications I have. So most of them are still sitting on the shelf. But that's not true anymore with the work that we do in this business. It is a very careful scrutiny. And so therefore we must teach our students how important it is to document every aspect of their work. And we spend a lot of time emphasizing that specific aspect. And even when we fail, or even when we follow this EPA protocol exactly to the limit, very often we fail to note that there are sources of error which can enter into our procedures. One of the campaigns that I have been writing on this last few years is the fact that we're dealing with a situation in the universities across our nation where many of our students are not exposed to field activities prior to graduation. So they graduate thinking that they're going to be modelers and thinking that they're going to be solving using Lenny Conoco's MOC model, and they're going to be the center of all the activity. And as it turns out, they're in the field drilling monitoring wells for the first 2 or 3 years. And many of these students have not actually seen the equipment that they need to use. So it's a very disturbing circumstance. Now, there are lots of things that I can say about that. One of the things that bothers me most is that they're not introduced to that process of discovering where error enters into their sampling process. For example, we have been doing a sample collection in a variety of sites around Arizona. We follow as precisely as we can the necessary procedures. Recently, we got back to the laboratory and we were reprocessing some of our samples and trying to backflush them through a column. And one of the things that happened, just totally unbeknownst to our expectations, was the fact that we were bleeding, so to speak, plasticizers from a little segment of tubing in our peristaltic pump back into our system, which had a higher concentration than the organics that we were interested in separating from our system. So it's right down to the wire. You go following all the procedures. But if you're thinking about sources of error, you'll double check and you'll triple check and you'll rub blanks. And when you do, you sometimes find that there are new sources of error that you hadn't anticipated. The second thing that I want to talk about is the analytical aspects of what we're doing, the chemical analysis, both field and laboratory analyses. We are dealing with an expensive business. It's very difficult for us to be really precise with our analytical work in the field. And there is a move. I have done an awful lot of consulting work myself. And there is this move to go from field to laboratory. The 25-degree clean room environment in the laboratory is so much better a place to do our analytical work. Unfortunately, the unstable parameters, so many of the volatile compounds that we deal with, must be preserved properly in the field. Now, this is especially a problem for students. And of course, here at the University of Arizona, we have a tremendous number of graduate students. And the thing that concerns me is that not only do they not go to the field and learn field experimental techniques, they're not introduced to instrumentation. So they're not aware of the error and uncertainty that can happen in the instrumental analysis. And so they don't really understand the need for field preservation and the need for very careful analytical work in the field. NSF has come out with a report that says that our universities are essentially in very abysmal shape right now because there's not capital available for the appropriate instrumentation to upgrade and to become state-of-the-art kinds of institutions. As a result, our students are moving more and more into this area of modeling and theoretical application and moving further and further away from field analytical work and laboratory analytical work. Now, the consequence of that is when we find ourselves in the field and it's 110 degrees and you're a long way from where you would like to be, you tend to find yourself looking for ways to finish the job a little faster. And I've done some work like this where I have to haul everything to the field. Many of you have done this very same thing, where you have to fly into a site and you bring your chem wipes and your plastic bags and everything that you need, your reagents and your third and fourth backup pH electrodes. Because you know that once you get there and do your work, it all has to be done at one site. And that's all. There's an effort, there's a time, and there's an expense problem in that particular aspect. And if this has not been ground into your thinking, how important this error and uncertainty aspect of the chemical analysis in the field is, you begin to take those shortcuts. You begin to take those samples and you don't document properly and you move back to the laboratory environment. And as a result, the quality of our data, which we need for the interpretation, is degraded substantially. And we take some of that responsibility in the universities, but we need to let, I think, the entire industry know that we are being financially hampered. So this is, it's a dirty job, and students are not particularly used to that. We teach a summer field course in the summer, and it's 110 degrees in May and June in Tucson, and so it's sometimes a difficult environment, but they need to learn this before they learn it on the job. Now that's introduction. I just wanted to talk about this aspect of qualifying our data before we get into the interpretive phase. The interpretation. I get feedback from students. They interview with a lot of the companies that are represented here. And the thing that everyone seems to be looking for today is a modeler. We want a modeler. If you can put model somewhere in your resume, it moves you up about 11 notches on the scale. Now, what do we model? What's this concept of modeling? I have identified here 3 model approaches, and I want to talk about each one of these. First of all, the transport modeling, secondly, chemical modeling, and thirdly, conceptual modeling. Now, when we talk about modeling, these 2 major areas I want to break down for you. The solute transport modeling, and there's equilibrium and mass transfer modeling. Now, there's a difference between the two, a major difference. In solute transport modeling, which is, of course, the rage, we are coupled to flow. Now, that's a very significant concept, because even though we're looking at the movement of a solute, that solute's migration is related intrinsically, mathematically, rigorously to the movement, the physical movement of the fluid itself. Now, the solute transport equation, as we're familiar with it, looks at that change in concentration with time as it relates to 2 specific terms, typically the dispersion term. How is it mechanically mixing along a flow path? And it's related to the velocity. Now, if we don't know those 2, then the aspects of transport are really reduced in their value. Now, there's a different kind of modeling which has become far more popular in the last decade, and I see it as something that is going to be incredibly useful in the future, which is decoupled from flow. You do not necessarily have to know mathematically and rigorously the velocity field in your system, nor do you have to know dispersion. You're looking at a system answering perhaps different questions. There are equations such as this which govern those kinds of processes. The first one would be a mass balance expression, where you're interested in maybe the total concentration of plutonium in your system. So you're working at a high-level waste site, or you're working at Rocky Flats in Denver, and you're interested in the distribution of the total mass of some given constituent. Then there's the concentration of the individual ion, and then all the complexes that form with that ion. Then there is the thermodynamic expression, which describes its activity in the system. The activity of the ions in the system as they're related to an equilibrium constant, a fundamental thermodynamic parameter. The third kind of equation that we need is the distribution of ions between the solid phase and the solution phase, where it may be a gas solution interface, or it may be a solid solution interface, a Henry's law constant of some sort, or a solubility product constant. All of these equations are essential for doing mass transfer kinds of calculations. None of them are rigorously coupled to flow. Now let's look at just a couple of examples. This is a slide of a cross-section of a recharge project that was done in Texas. Warren Wood and Rich Brown and a variety of others were involved in this project. And we were interested in the movement of solute through the unsaturated zone from the surface to the groundwater. Now, we ponded water on the surface, so there was a berm constructed, and we had a source of water of one composition, which was different than the water composition in the groundwater. We had a variety of lysimeters that were buried all through the section. We knew the geology very well, well-characterized mineralogy, X-ray diffraction work, geophysical logs, Warhol geophysics, you see a gamma log there, well-characterized cross-section. The question was, how do you detect the chemistry along the flow path, and what can you do in the way of modeling that system? Now, one of the constituents that I was particularly interested in was fluoride. And we've published the work here on the fluoride geochemistry. In the high plains of Texas, fluoride is really a high value. You can get 5 or 6 milligrams per liter of fluoride. And the concentrations of fluoride can be just contoured. If you contour just looking at the individuals who live in the high plains, their intensity of modeling on their teeth, the brown teeth, you make a very nice contour map of the concentration of fluoride that's in the groundwater. So fluoride is a hot topic in the high plains of Texas. So here's our one-dimensional transport model. And we're interested now in the partitioning between the solution phase and the solid phase. How does fluoride partition? Now, you can use a simple expression like a Langmuir isotherm. There are many isotherms that you can use, but we used the simplest because it models the experimental laboratory data very well. So here's what we get. Here is a model for the movement of fluoride from the surface to the groundwater through that intermediate vadose zone. And I have two plots here. This is, if you look at the yellow, that's the observed data, the experimental data. We had points, many points for every day. You look at the time period April through May, so it's a 30 days worth of activity. And then we have the computer model. We simulated that with a one-dimensional transport model. And that was really a nice fit. Now, I was overlooked for the Nobel Prize that year, and I suspect the reason for that was because we only looked at 0.6 of a meter. So that wasn't a long transport distance, but we were able to very successfully model the breakthrough of fluoride over a very small distance over a long period of time. Now, the point is that we were looking at input values here. This is a graph showing the input value of fluoride, about 1 milligram per liter. And this is the section that we modeled 0.6 of a meter down, and we were able to model very precisely this removal of fluoride. And this was not in a soil zone. This is actually down into the vadose zone because we'd scraped away the soil. Then we went to the 2-meter depth, which is just 1.4 meters further down, and we see a totally different kind of response, unexpected response. Thinking only of what we call the near wellbore conditions, if we had made our projection further down, we would have not ever seen the fact that there is a tremendous source of fluoride here in this groundwater system, and that this tremendous removal that we see here, almost complete removal of fluoride for over 30 days, is relatively insignificant as you get 2 meters away from this particular zone. So our projected modeling, our long-term distance modeling using near-field effects was not as good as it could have been. Okay, that's a very simple expression. That's a Langmuir isotherm. That is the simplest isotherm that you can use for adsorption. What about a more complex chemical reaction? These are probably the 3 most commonly used expressions for ion exchange. And once you get away from something as simple as the Langmuir expression, which is a theoretical expression, you can derive that from first principles, and move into this realm of more complicated chemistry like ion exchange, it becomes totally empiricism. These equations, of course, come from the soils literature, and they are useful for describing monovalent divalent exchange. So you can see the Davis, the Van Slow, and the Capone equation are typical equations which are used for this kind of approach. Now here is a model that was published by Grove and Wood in groundwater a number of years ago for this very same site. Now this is the same data set. Now what they've done in this particular series is they wanted to model this ion exchange process, but they chose first to do it in the laboratory. So they determined their equilibrium constants for sodium-calcium, magnesium-calcium exchange in the laboratory using the data for a column experiment, but the samples, of course, from the field. They were able to model really very well this whole process of ion exchange of calcium, sodium, and magnesium in a column experiment. Then we looked at the actual field data. Now you notice that this is for the 2-meter sampling depth, which is where we had trouble before with fluoride using simple Langmuir exchange. And you can see how well this fits. This is really a very good fit. The standard deviations are very tight. For an ion exchange reaction in the field, this is really quite good. But now you go back. Now see, we calibrated at 2 meters. It fits very well at 2 meters. But you go back to the 0.6 meter, where I had all my incredible success, and you find that the same parameters that we use at 2 meters do not fit the situation that you see at 0.6 of a meter. So you see there is already, in this very short distance, over very short time frames, relatively simple reactions, there is a problem of scale and a problem of aquifer heterogeneity. Now, I want to go just one more. Another major tracer experiment that we've done on the high plains of Texas, the data are now being processed, have several students working on this, is looking at a different circumstance where we inject a whole series of tracers down the wellbore and monitor the change in the concentrations of a variety of ions horizontally, radial flow from the central well. Now, excuse me, we have a large number of analyses, and I just want to highlight with a few slides the same kind of phenomenon looking at radial flow horizontally. So here's our injection well. We have a variety of sampling points in wells that are spaced from 2 meters, 5 meters, 10, 15 meters, and 30 meters away from this injection well. Multi-level, so we have essentially 4 sampling points in any given well, except for these continuous wells, as we call them. And we're looking at a number of locations radially distant from that injection well. This would be a typical multi-port a sampling nest. So there's a variety of samplers, and the samplers are denoted by color. This would be the green, orange, red, and black samplers, and easier to take care of in terms of descriptive slide presentations. This would be what one sampling device would look like. So it has a screened interval, so you can sample essentially about 15 centimeters of section and grab that sample and bring it to the surface. So we did a series of tracer tests, and I just want to focus today on some bromide, and you'll hear a lot about bromide tomorrow. So one test where we're looking now 2 meters away from our injection well. And here you see represented, the data points represent an actual chemical analyses, and the solid line represents our attempt to model it. The thing I want to point out is that the shape of the curve, just remember the shapes. Don't worry about the parameters. All these parameters refer to flow, to porosity, to thickness parameters, to diffusivity, changing the characteristic length. There's a variety of parameters that you know that we can use in our transport models to describe this process. What I want you to see, though, is what happens in a very qualitative sense in this system. So here you see the data. It's a relatively narrow peak. The breakthrough is relatively fast. I do want to point out the tailing that you observe here. This is bromide, which is our, quote, ultimate conservative tracer. And there is a substantial amount of tailing in this curve, which I think needs to be explained. Okay, this is at 2 meter orange. This is the highest level that we considered. Now we're going to go down 2 meters away to what we call the 2 meter red. Same radial distance, same geology. If you look at the logs, this is essentially an indistinguishable zone from the zone that we call the orange zone right above. And notice the difference. Much broader, much shorter, and the tailing is not quite as significant when you get out in this range. Now we'll go down one more level, which was the zone we call the black zone, and on the logs you do see that it is a little bit tighter, and we essentially have, throughout the entire experiment, almost no breakthrough of tracer. So here we have 3 specific zones that we looked at in detail for the conservative tracer bromide, 3 different behaviors, and then we have a well at 5 meters that we want to look at. Now, notice that we have relatively narrow, also some tailing, and we've modeled this with a variety of different characteristic lengths. Now, this is the red one, halfway down. Now, this is, I don't know if you picked this up, it's hard with all these slides. It's an opposite response as what we saw before. Here we're seeing a very narrow, very fast kind of behavior, so that we're getting to the 5 meter well at this zone faster than we did at the zone above it. Whereas if you go back to the 2 meter well, this particular zone was the slower zone and the one that had the fat, short kind of breakthrough curve. So we see moving horizontally from 2 meters to 5 meters, on the first 2 meter well, we have now reversed the arrival times, moving horizontally from our injection well. Now, the interesting thing is, we go now one step further as we look at a continuous well, one well that's open through the entire section of the aquifer. We're sampling all 3 of these zones, and we end up with a very fuzzy-looking, fat-tailed behavior, which does not look anything like the individual zones that we looked at before. And now this is the 10-meter continuous well, and you can see the effect of radial dispersion away from the well. Integrated effect of the other 3 tests. Now, let me just show you one other thing, and I'll finish my discussion with this. We then conducted another tracer test, same system, using 4 tracers, the halides, of course, that we're all interested in. A pulse only lasted 2 minutes. Now, here's what the breakthrough curves look like. This would be at the 5 meter orange zone. Now, I want to point out the difference in arrival times. Now, we're not plotting C over CO. We're just plotting absolute concentrations here because we wanted to observe fluoride. Fluoride has a lot of reaction, as we already have talked about, along the flow path. But if you look at this, iodide, bromide, chloride at 5 meter orange have almost identical behavior. They break through all about the same time. Fluoride, of course, has different reactions, so it behaves differently. Now, I want to go to 5 meter red. Look at the difference. If you look at the logs, the gamma ray logs, if you look at the core, and if you look at neutron logs, if you look at any other method of comparison for just strictly aquifer matrix comparison, they look very similar. But there was almost an instantaneous arrival time at the red zone, whereas there was a much delayed, several-hour delayed time, 5 meters at the lower zone. Now, why in the world do we want to talk all about these kinds of things? Here is the continuous sampler, which integrates all of those effects. So you see early arrival time for some, and then you see a later arrival time for different zones, and then it tails off. The reason I want to talk about that is because this is the kind of data that we deal with in our predictive modeling. We typically have near wellbore data. We have a pumping test which samples a small region. We have core tests from our laboratory studies. We try to make some sense out of our geophysical logs or our cores that we've taken back to the lab. And from that data, we are now required to do this kind of modeling. We go from that small scale, 0.6 meters to 2 meters, and we have to make extrapolations like this. Here's the real field site we deal with, the contaminated zone. We're looking at tailings pile, and we want to know the question of how far and how fast. And we have a contaminated environment. Maybe it's sulfate, maybe it's uranium. but you're looking at the distribution of some species between some tailings pile and the river. And the EPA or the local regulatory agency says, how long will it take? What will happen if we do this? So we have to make this predictive model. So from this one-point calibration, which is what we have now, we've drilled a few monitoring wells to kind of know the distribution of the species. We have some idea about the source term. We now have to make the prediction of what's going to happen in 25 years, 50 years, and 70 years. And I just keep thinking, I can't make that prediction very well if I remember the number of tracer tests that I've done and that 0.6 meter, the 2 meter, the 5 meter, the 10 meter kind of projections that I'm making, and the difficulties in the aquifer matrix dealing with those heterogeneities. Now, why am I bringing all this up? Most of you follow the literature, and most of you are aware of the comments that are being made in groundwater, water resources research, monitoring review. The kinds of comments that we're talking about now are, we're in trouble with our transport modeling because we are publishing diagrams like this. There's no error on our diagram. We are willing, with all our audacity, to say this is my prediction for the distribution of sulfate 50 years from the future. As a result, we have a progressive disbelief of our transport modeling. In fact, if you look at the industry as a whole, I think there is probably very little belief in the transport modeling which results in these kinds of diagrams. So we have now the scientific community which is saying, I am really in trouble with my predictive modeling. I must come up with a method for error and uncertainty. Unfortunately, we still have the private sector, for which I have been a part as well, that is continuing to do this kind of modeling. So if we're going to regain any level of credibility, I think we have to start in the classroom, but we also have to start in our scientific communities. I think if you've noticed some of the symposium that are scheduled for the next few years, one of them early next year, deals with this very specific issue of error and uncertainty in transport modeling. We need to be able to put together a method for describing the minimum and the maximum. Just like I showed you with our Stanton experiment, the first time arrival time. What is that first arrival of the constituent? And what is the maximum arrival time? And what is our tailing going to look like over time? We need to come up with techniques for doing that and no longer publishing these kinds of diagrams. Because we know, we look at the literature, look at the jobs that you did 15 years ago. Are your predictions holding up? So how do you do it? There are, just want to make 3 quick comments. There are 3 things that contribute to this uncertainty. The conceptual model, the mathematical model, and our data. First of all, the conceptual model. Very often we're having trouble with this very first step. We don't consider the fact that it's fractures as opposed to porous media, or it's a single porosity model versus a double porosity model. Our concept is wrong. Very often our mathematical model is wrong. We have oversimplified our system. I think there is a tremendous difficulty in modeling systems now where we simply use an analytical approach or a one-dimensional model to try to model 2-dimensional and 3-dimensional systems. The heterogeneities, the scale of the heterogeneities is greater than the scale of our sampling point. So we're using the wrong kinds of mathematical structures. But I think even more important is this one area here, the data itself, our physical parameters. We're not taking care of the dominant parameters in our system. And I think one of the most significant parameters with which we have to deal is this issue of hydraulic conductivity. The second thing is our chemical parameters. We're not identifying our species properly. We're not speciating. We're trying to transport the wrong species. And then this whole thing of spatial variability. How do we deal with that? There are basically 3 ways that we can handle this. We can actually build a field scale experiment and do a tracer test, the actual size of the question that's being asked. Somewhat impractical. The other thing is to do a whole series of small scale experiments and take that data and try to extrapolate it. Another is to use a variety of different kinds of data types and then use a geostatistical technique like Kriging, for example, to try to make some sense about the internal structure of those points. So a geostatistical method. And another more popular method, I think, now is using stochastic techniques, looking at the probability distribution of the heterogeneities in your aquifer. But the point I want to make is that if we are going to regain any sense of credibility in this whole business of predicting the movement of contaminants in the environment, I think we have to move to the point where we are putting error bars and uncertainty on all of our models. Okay, Jim Ye, I have a reference there. Jim Ye has a nice discussion of the different kinds of stochastic models that are available.